All right, now that we've talked about how hormones are made and the different kinds of hormones, let's talk a little bit about how they work. So right now we're going to be talking about hormone receptors. And the reason I went over in the last lecture a lot about whether hormones could or could not penetrate cell membranes was because that makes a big difference as to where their receptors are and what those receptors tend to do. So let's set this up. We'll put sort of the outside of a cell here. And we'll imagine hormones are coming from outside toward the cell and then talk about what they do in here. So in here I'm going to have the nucleus, where we've got the DNA. Now, some hormone receptors are what we call intracellular. So for an intracellular receptor, that receptor could either be here in the cytoplasm or even here in the nucleus. Now, if you've got a hormone that can penetrate cell membranes, so let's make one of those hormones here. This would be, for example, let's say this is a steroid. Steroid hormones, since they can diffuse through cell membranes, can just pass right into the cell and then either bind to a receptor here in the cytoplasm or go straight to a receptor in the nucleus. If it binds to a cytoplasmic receptor, that receptor then moves to the nucleus usually. And these steroid hormones, their mechanism of action is usually to bind to a receptor, which then binds to DNA and changes which genes are being transcribed. So steroids are almost switches to change which proteins is this cell making. That can be a profound change. That can really change what the cell does at a very deep level. But their receptors are usually inside the cell because they can pass through these cell membranes. Compare that to other receptors. Okay, so another lipophilic hormone, like a thyroid hormone. Thyroid hormones have to have some kind of transporter. It's not a receptor, though. That transporter moves them into the cytoplasm, and then in there they will usually find a receptor, which then enters the nucleus like a steroid. And their mechanism of action is also like a steroid. Usually their receptor will bind to DNA and change which genes are being transcribed. So the main difference here is that thyroids have to be transported across the cell membrane, even though they're lipophilic, whereas steroids just diffuse across the cell membrane and bind to receptors. Now, one interesting thing there is that for a steroid, if it can diffuse in, it could just diffuse right back out, too. So when it diffuses into the cell, if it does not find a receptor, if this cell does not have a receptor for that steroid, then that steroid will tend to just come back out, get onto another plasma protein, and move on. Whereas thyroid hormones, once they are transported in, they don't easily leave the cell. Steroids, once they bind to the receptor, they won't leave, because that receptor then takes them to the nucleus where they're going to do their job. Now, that's for hormones that have receptors inside the cell, these intracellular receptors. Intra meaning within. Other hormones like catecholamines, as it turns out, melatonin, and almost all peptide hormones, those usually use cell surface receptors. So these, which are hydrophilic, and in the case of peptide hormones usually large, cannot cross the cell membrane, which means these kinds of hormones have to bind to a cell surface receptor. They never actually enter the cell. And the cell surface receptor will produce some kind of second messenger, which will then have some kind of effect in the cell. Usually, not this kind of thing. Usually not altering the DNA, but some other kind of effect which changes what the cell is doing. Often, that will affect what other things the cell secretes. Or it'll change what kind of other receptors the cell will put on its surface for other things. So, for example, one of the effects of an insulin receptor is to cause the cell to put out more glucose transporters on its membrane. These effects of cell surface receptors are usually relatively fast, taking place in seconds, minutes, hours, compared to the effect of these intracellular receptors, which can take hours, days, weeks to really have a big effect. So these are more rapid changes for the things that can't penetrate the cell. Now, as it turns out, some of these things, like steroids, like for example estrogen, some of its receptors are cell surface receptors, so it actually can do both. Some steroids have both intracellular receptors in some cells and cell surface receptors in other cells that produce different effects. So that's one way that a hormone could have more than one kind of effect in different cells, or even within the same cell. So, one thing I want to note here is that all of the cell surface receptors use second messenger systems. So these are either going to be G-protein-coupled receptors, which you've learned about, or receptor enzymes. These two kinds of receptors that use second messenger systems. So the next thing we're going to do is talk about some specific examples of hormones and their receptors and some of the effects they have. Actually, before we go on, I did want to remind people of a couple of things about those second messenger-based systems. So we're going to look at several common types of these things. One of them is going to be G-protein-coupled receptor, which is coupled to an adenylyl cyclase. Another is going to be a G-protein coupled receptor which is coupled to a phospholipase C. So two different G-protein coupled receptors which are attached to different enzymes. So just a really quick overview and a little more information about what those do. So if you remember, adenylyl cyclase is an enzyme that turns ATP into cyclic AMP, also known as CAMP. Cyclic AMP is a very common signaling molecule within cells. And one of the ways that it often works is by affecting protein kinases. So protein kinases are enzymes which add phosphates to proteins. So often the way this is going to work is that activating this kind of G-protein coupled receptor will cause it to start making cyclic AMP, which will then influence whether protein kinases are phosphorylating other proteins, usually enzymes, which are then making third messengers. So that's part of that signaling cascade. It is also possible that what happens when you bind to this receptor is that it turns off the adenylyl cyclase, which then stops making cyclic AMP, which then has a different influence on protein kinases. So, just so you know, it can go either way on that one. The phospholipase C worked slightly differently. So if you remember, phospholipase C turns membrane phospholipids into two different second messengers. One of them called DAG, diacylglycerol, and the other was IP3, inositol triphosphate. IP3 usually works by opening calcium channels deeper in the cell. So IP3 usually causes calcium release. So it'll let calcium out into the cell from internal storage. We've seen some of that before, which then activates calcium-sensitive machinery within the cell. Excuse me for a second there. DAG DAG often also influences protein kinases. So it can have some similar effects to cyclic AMP. Okay, hopefully that summarizes that just a little bit. These are the G protein coupled receptors. Some of these things also work through membrane enzymes. They may, for example, bind directly to guanylyl cyclase or something like that, but we'll talk about a few of those as well. Alrighty, I wanted to share with you a few examples of how some specific hormone and receptor complexes work, so you can relate this to stuff you already know about. So just for an example from the urinary system, let's talk about the vasopressin receptor. And here we're specifically talking about the vasopressin receptor V2, which is the one found on collecting ducts. So vasopressin is a peptide hormone, meaning it is hydrophilic and does not cross cell membrane, so this is going to have a cell surface receptor. In this particular case, the V2 is a G protein coupled receptor linked to an adenylyl cyclase. And so that takes ATP, turning it into cyclic AMP. And in these particular cells, cyclic AMP triggers fusion of vesicles. So if you remember how these cells worked, if this is our apical membrane, remember we had those vesicles with aquaporins on them, that cyclic AMP is going to be the thing which makes these vesicles come up, fuse with the membrane, and make this cell permeable to water, which allows us to reabsorb water from the collecting duct. So that's just an example of how this is how vasopressin works. Binds to the V2 receptor, which activates adenylyl cyclase, which makes cyclic AMP, which triggers the machinery that causes these vesicles with aquaporins to move up and fuse with the apical membrane. So an example right there. Okay, here's another example for you. So we've talked about some of the adrenergic receptors, the ones that respond to norepinephrine and epinephrine, for example. Here's a couple of them, the alpha-1 and the beta-2, and a little bit about how they work. So, for example, alpha-1 receptors, you may remember, respond to norepinephrine, and they tend to cause the contraction of smooth muscle that they're connected to, like, for example, in blood vessels. So the way that's going to work is if we imagine that we're looking at a smooth muscle cell here, the alpha-1 receptor is a G protein coupled receptor linked up to phospholipase C. So, when norepinephrine binds to this G protein coupled receptor, it activates the phospholipase C, which makes the second messenger IP3. And IP3 causes the release of calcium from inside this cell, that's Ca2+. And remember what calcium does. Calcium binds to calmodulin, which activates which phosphorylates the myosin light chain, which activates the myosin light chain kinase, which then phosphorylates the myosin and causes the cell to contract. So, norepinephrine activating the G protein coupled receptor, alpha-1 receptor, which activates phospholipase C, which makes IP3, which opens the calcium channels, which lets out the calcium. Now, compare that to the beta-2 receptor. So now imagine that we're looking at a smooth muscle cell, but this one has a beta-2 adrenergic receptor. The beta-2 receptor is connected to adenylyl cyclase. So, in this case, it responds better to epinephrine. So epinephrine binds to the receptor, which activates the adenylyl cyclase, which makes cyclic AMP. In this case, cyclic AMP activates something called protein kinase A. So protein kinase A, when it's activated, phosphorylates myosin light chain kinase. You remember myosin light chain kinase. That's the enzyme which adds phosphates to myosin. However, in this case, when protein kinase A puts a phosphate on the myosin light chain kinase, that phosphate causes it to change shape in such a way that it actually can't do as much work on it can't phosphorylate the myosin as well. So here, we're adding a phosphate to the kinase, which acts as an allosteric inhibitor. You add that on, the kinase changes its shape, and the active site no longer works as well to add phosphates to myosin. So let's think about how that's going to work. Epinephrine activates the receptor, which activates adenylyl cyclase, which makes cyclic AMP. Cyclic AMP activates protein kinase A. Protein kinase A adds a phosphate to myosin kinase, which makes it not work as well. If I can't add phosphates to the myosin, then the cell can't contract, which is why beta-2 receptors tend to cause smooth muscle to relax. They interfere with the mechanism that would cause the cell to contract. They make it so we can't phosphorylate the myosin, so the myosin phosphatase ends up removing the phosphates from the myosin, and we end up with the cell relaxing. So, 2 examples of how these hormones can affect receptors, which then affect smooth muscle through 2 different G-protein coupled base mechanisms. Thought it might be kind of interesting. So now that we know a little bit more about how these hormones have their effect, let's now talk about how we get rid of them. So hormones, like neurotransmitters, we don't want to be active indefinitely, which means we need a way to terminate their signal. And how quickly that works depends a lot on the hormone. Some hormones don't last very long. Vasopressin, for example, has a half-life in the body that's measured in, I think it's in minutes. That once it's secreted, it's removed from the body very quickly. Others can last for days or even longer. So let's talk about some of the mechanisms by which they're removed. There's 4 main ones. Hormones can be removed by excreting them from the kidneys, that as our blood passes through the kidneys, the hormone may be one of the things that gets filtered or even secreted, and then it ends up in the urine if we don't reabsorb it. So that will gradually remove it from the body as if it were a metabolic waste. Another is that the liver often has enzymes for breaking down hormones and other things in the blood. So we may secrete some hormone into the blood, but then the liver gradually removes it, so that unless we keep putting it in the blood, the levels in the blood will then drop as the liver breaks it down. Another one is that the liver usually might put enzymes into the blood which will break down the hormone when they run into it. So rather than waiting for the hormone to come to the liver, we send something out after it to break it down. There's a kind of an interesting wrinkle on that one, which is that sometimes the enzyme doesn't just recognize the hormone at its active site. Sometimes the enzyme will recognize the hormone when it is bound to a cell surface receptor. And think about how that one's kind of interesting. Let's say I am the circulating enzyme, and this cap is the hormone. It may be that my active site doesn't recognize the cap. So if I'm circulating around and I run into the hormone, I say, nope, you're not what I'm looking for. Now let's say this is that hormone's receptor. So hormone binds to receptor, and if the enzyme finds that, that's what it recognizes, so that's what it destroys. Now the implication of that is that if I break down the hormone wherever I find it, then that hormone won't last that long in the blood. But if I have to wait until it's bound to its receptor, then I know that that hormone molecule will have affected at least one receptor, and it's less likely I'm going to find them bound together. So that will extend the hormone's life and make it last a little longer and have a bigger effect. Some hormones are like that, others just get broken down by circulating enzymes. Finally, in the case especially of steroid and thyroid hormones, they are internalized into their cells. When they enter the cell and find a receptor, they then stick to that receptor and they don't usually come back out of the cell. So that's a way of removing it from the blood. I put it in the blood, and when it gets to its target, it disappears into the target, so then it's no longer circulating and it can't affect any other cells. Those are some mechanisms by which we can terminate hormone responses. Now there's one last topic for this first lecture, which is a very brief looking at some mechanisms by which we control when hormones are secreted. In general, so let's talk control of hormone secretion. Now, much of the time, we've got a gland or a cell that produces the hormone. Now, one question is, how do we know when to produce the hormone, and how do we know when to stop producing the hormone? Usually, there's going to be some kind of negative feedback system involved. Sometimes, the hormone itself is a signal to the cell to stop secreting the hormone. So we make the hormone, and then when we see the hormone, we say, okay, I don't need to make any more hormone. That would tend to result in a small but steady amount of hormone in the blood. Another possibility is that the hormone causes some sort of effect. For example, that might be to produce another hormone, in which case that second hormone might be the negative feedback mechanism. So you'll see this, for example, when we talk about the hypothalamus and pituitary. The hypothalamus makes a hormone that tells the pituitary to make a hormone, that tells another gland to make a hormone, which then tells the hypothalamus to stop making that first hormone. So we have this sort of multi-level feedback mechanism. In any case, usually this is going to be based on some kind of negative feedback mechanism that will make the hormone, and either that hormone or some effect of that hormone will tell the original gland not to produce the hormone. Sometimes there will be something else that tells it to produce the hormone. There might be a signal coming in from somewhere else that says, yes, produce more of that hormone. That's maybe how this gets started. This ends up being very specific to each hormone. So in the last lecture here, we're going to talk about some specifics of how certain hormones are controlled by negative feedback and mechanisms like this. We'll go with several possibilities. One of them will be a very simple negative feedback system. That'll be for parathyroid hormone. One will be a more complex, sort of two-sided negative feedback system. That'll be for insulin and glucagon. And then we'll talk about what's called the hypothalamic pituitary axis, which is a common way that we regulate hormones in a multi-step, complex negative feedback system. So we'll get to that in the next lecture.